So for this lecture, I actually kind of combine prokaryotes and eukaryotes as kind of a similarity or compare and contrast. And that's actually one of your objectives, right, is to be able to compare and contrast prokaryotes versus eukaryotes. So as we talk about some of these structures, it's a good opportunity to say, okay, what does a prokaryote have versus what does a eukaryote have? And how are these things linked? What do you think is similar about what a prokaryote has versus a eukaryote? I'll give you an example, a ribosome. What does a ribosome do? It creates proteins. Are the ribosomes that are found in prokaryotes and eukaryotes the same? No, but they're similar. And definitely the major similarity is that they both what? Make protein. They both make protein, right? So, you know, the commonality a lot of times is going to be function, but how they achieve that and what they're made of is going to vary a little bit. So again, we'll look at different drawings of prokaryotes versus eukaryotes and look at these similarities and differences. So again, tables are a good way to organize information, especially when you're looking at comparing similarities versus differences. So I put our little bacteria on its side here, our little prokaryote representator, to be able to squish it on. And then we have our two example eukaryotes that are microscopic, right? So this is a protozoa cell, maybe I was trying to think of an actual example of one that has a flagella. Something like Giardia, for instance, right? Or a fungal cell, in this case it's a yeast cell. Notice that for eukaryotes, what's the defining characteristic that makes both these cells eukaryotic? The nucleus. So they both have a nucleus. They also both have a region within the nucleus referred to as a nucleolus, something that we're going to talk about. Not an organelle, but again, notice I said region. And then, of course, eukaryotes have other membrane-bound organelles like mitochondria, right? Endoplasmic reticulum. What makes the rough endoplasmic reticulum rough? Ribosomes. That's where protein synthesis of secreted proteins happen. So then another place where things get processed, a whole bunch of membrane sacs that are stacked up on each other are known as the Golgi apparatus, right? A lot of this should be reviewed from biology class, right? Some eukaryotes need specialized vesicles like water vacuoles. Paramecium has contractile water vacuoles, right? Because again, they live in an aquatic environment and water continually rushes into them. So theirs actually contract and squirt the water back out, right? So they always look like they're winking at you. They're so cool. Others may have storage. The important thing is for these guys is all these things are membrane encased, right? You have lots of these membrane compartmentalizations, these organelles that exist in eukaryotes, and is one of the defining characteristics for them. Of course, they have ribosomes. All cells have to have ribosomes. These are not organelles. Some people get confused and think it's an organelle. It's not membrane encased, right? But they're scattered throughout the cytoplasm on the rough endoplasmic reticulum. They're even sometimes associated with the membrane. And so, as we know, these are used to make ribosomes. They're very large in comparison to most molecules in cells. They're very large molecules, right? And so for the most part, if you were looking at an electron micrograph, you know, a picture made by an electron microscope, they look like little dots in the cytoplasm. And so again, the little dots that you see here in these drawings represent the ribosomes. Now in contrast, right, you do have DNA inside a prokaryotic cell, but is it encased in a membrane? No, right, because they don't have organelles. That region where the DNA and the proteins that complex with it and the RNA that's associated with it, that region is typically referred to as the nucleoid. Again, not to be confused with the nucleus. And then common features that you'll find in most prokaryotes is, of course, they're going to have a membrane. All cells have got to have a membrane. But then they'll probably have a peptidoglycan layer, right, that cell wall layer. And then some of them, as we know, have an outer membrane, and even still some of them have what's referred to as a capsule, something that we're going to talk about soon. Here they're also representing lots of little hair-like appendages known as fimbriae. This helps them attach to surfaces, and for some of them even provides a little bit of motility. Although motility is primarily done by flagella if a bacteria is motile. Of course, one of the exceptions, of course, is the spirochetes, right? Where they have the axial filaments and they're allowed to corkscrew through their environment. And then, of course, they're showing a flagella here for our eukaryote. But their flagella is made up of different stuff and it moves differently than prokaryotes, as we'll see in a moment. So again, same function though, right? Motility. But structurally they achieve it differently, and even how it moves is different. And then unique to eukaryotes are little tiny hair-like appendages, which are basically flagella, but they're smaller flagella. Anyone know the name of them? Paramecium is covered in them. Cilia, right? Cilia are only found in eukaryotes. So the cytoskeleton, as the name implies, right, it's a skeleton within the cell and it helps support the cell. When I was sitting in your guys' seats, we didn't talk about prokaryotic cytoskeletons. We didn't know they existed. We didn't have the technology available at the time to know that prokaryotes had a cytoskeleton. But if you think about it, it makes sense, right, to have some type of structural support inside of the cell. Now, of course, the cell wall is very supportive for bacteria and archaea, for prokaryotes in general. But they have found, right, as shown here, some proteins specifically inside the cell that help with cell division, just like our cytoskeleton does for our cells, for eukaryotes, that help localize certain proteins to particular areas, right, so it helps in the organization within the cell, and helps determine that cell shape, which is paramount for eukaryotic cells, right? Eukaryotic cells, it's really all about the cytoskeleton because we don't have cell walls. But for eukaryotes, the cell wall, but the cell cytoskeleton also contributes to its shape. So some eukaryotes have additional functions, right? It's a much more complex cytoskeleton, right? We have three major components that we talk about when we talk about the eukaryotes. We talk about microfilaments. So in addition to, helping with cell shape and localizing things in cell division, these guys help in cell motion, shape changes, moving around, things like pigment granules, and especially in amoeboid cells, so like in amoeba, how they're able to push their cytoplasm and move, right? That is made possible by these microfilaments. These microfilaments are actually what's pushing that membrane and the contents of the cytoplasm so that they can move. The protoplasmic streaming that's seen in slime molds, where you'll see the inside of the cell move, is because of these microfilaments. Now, as the name implies, these are the smallest of the cell cytoskeleton features of eukaryotes. The next, as the name implies, are the intermediate filaments. They're intermediate in size. These are important for the nuclear lamina, so inside the nucleus. So notice that most nuclei have a particular shape to them, right? When you look at a nucleus of a eukaryotic cell, and those of you guys in lab right now, you even found that some organisms have a characteristic shape to their nucleus, right? Bacteria coli has this kidney bean-shaped nucleus. Although most of the time it's spherical, right? But there are some that will have these unique shapes to their nucleus, like Bacteria coli. This is made possible because of the internal intermediate filaments inside the nucleus. So it's almost like a scaffolding for the nucleus itself. Now the membranes that encase the nucleus, there's actually two layers of membranes. So it's actually referred to as the nuclear envelope. because it isn't just a single membrane, it's actually two membranes. And when you have organisms that are made of multiple cells, and when you have tissues form, these filaments help maintain stability between the cells that form tissues. Microtubules, again, help with shape, and they're involved with the microfilaments for cell movements and transporting stuff inside the cells. But then we also have specialized ones, like the mitonic spindle that you talk about during mitosis that pulls apart the copies of the chromosomes. That's a microtubule. It's a specialized microtubule. The cilia are made up of microtubules. So are flagella for eukaryotes. When we talk about inclusion bodies, these are found only in prokaryotes. And what they are areas where they're going to store stuff, right? It could be organic or inorganic in nature, and they could be protein or phospholipid and protein encased, but these are not considered membrane encased organelles, right? Because again, prokaryotes don't have organelles. But they're a way to compartmentalize and store stuff inside the cell. And again, remember, these are single-celled organisms. All prokaryotes are. So they have a whole lot of problem with osmotic pressure. And how much stuff you have inside your cell really factors into how much pressure you're under. So if you can take some things out of this equation, then it will help with the osmotic pressure. So the best example I've found to help you guys understand this phenomenon is, you've ever seen those plastic ice cubes? You know what I'm talking about? Like it's plastic, there's some type of liquid on the inside, and you put it in the freezer and you use it just like an ice cube. The difference is when you put those plastic ice cubes in your drink, do they melt? No, they melt inside the plastic, but that whatever substance is inside the plastic doesn't go into your drink. So unlike traditional ice cubes, which some people don't like to put in certain beverages because when it melts, what does it do to it? It waters it down. It dilutes it, right? So I like really cold orange juice, but I don't like to put ice in my orange juice because it waters it down, right? So it goes into solution. Well, these inclusion bodies are like plastic ice cubes. They keep the stuff on the inside, so it never becomes part of the cytoplasm, never becomes part of that solution that we call the cytoplasm. And the cytoplasm is made out of a lot of water. So remember, osmotic pressure happens because of what? Water movement in response to how much solute, how many dissolved substances are in that water, right? That's what makes it a hypertonic or hypotonic solution. Well, the problem is most cytoplasms are hypertonic. There's a lot of stuff in there, right? Lots of stuff. So water tends to want to what? Go inside that cell, and again, that pressure pushing up against the cell wall. Well, if you put the stuff in these little cubes, in these inclusion bodies, it doesn't factor into that equation of being hypertonic. You could almost make the cytoplasm, if you put enough stuff in inclusion bodies, you could make it isotonic. You can make it balanced with the outside. So that's a way, and just like most organisms, is food always readily available? You can eat whenever you want? No, right? A lot of animals, like dogs and stuff, they'll eat and eat and eat, right? They won't stop because they don't know when it's going to be available again. Microorganisms will take in and take in and take in because they don't know when they're going to see it again. They'll store it. So when nothing is available again, then they have stuff to eat, right? They stored it aside, like a squirrel hides nuts, because they're only available during the season. When winter comes, then there isn't going to be any food. Of course, that never happens here, but they still stash everything. It's instinct for them. They're still more plentiful, right, during a particular season. So that's one of the main reasons, right, is to be able to store but to store in such a way that's not going to affect the osmotic pressure of that cell. So for eukaryotes, do we have structures that are similar in function that allow us to store stuff? Yeah, give me an example. Lysosomes? Yep. Someone said something else. Nope. The water vacuole in the first picture, right, where you're storing water. So different vacuoles that store different things, but what's the key difference here between vacuoles and lysosomes as opposed to inclusion bodies? They're membrane-bound. And lysosomes are a special type of membrane-bound vesicle. They contain what inside of them? Acid and digestive enzymes. And this is why we keep them encased in a membrane, because we don't want them running willy-nilly through our cell and destroying anything they get their hands on. And that's what enzymes will do, right? We have to keep them under control. We only let them digest what we want, right? So we give it to them. So plasmids, which are mainly in bacteria, which we know are prokaryotic, right? But they have been found in some yeasts, which are eukaryotic. So although this is a main feature you see in bacteria, prokaryotes, it has been seen in some eukaryotes, but it's rare. What is a plasmid? Composition-wise, it's just DNA. The important thing here is that it's additional genetic information. So this is in addition to the chromosome. It typically is circular, as you see in the picture, and it usually imparts some type of advantage to that organism. So the scary thing for us in this world today is that several bacteria have acquired resistance plasmids. So these are plasmids that contain information on them that allow them to be resistant to antibiotics. And the really scary thing is they can give these plasmids to other cells. They can share this information. So it's not necessary that a cell has it, but it's very common in bacteria to have these extra DNA. And they'll actually even keep multiple copies of these plasmids in their cells. Why, I don't know. Other than maybe you can make more of the stuff if you've got more DNA, right? Because you've got to have the enzymes read it and the ribosomes make it. So you can make a whole bunch at once if you have more than one copy, right? You don't have to wait for somebody to finish at the copy machine, right? The RNA polymerase doesn't have to wait. He's got another piece of DNA he can go to and copy that. The chromosome, though, typically, if a cell has an extra copy of it, it's getting ready to divide. They don't keep extra copies of their chromosomal DNA in their cells. So for prokaryotes, I'll always refer to as the nucleoid, but in the literature you'll see other terminology for the region in which the DNA for a prokaryotic cell is housed. For the most part, it's usually in the center, like you see here in the specially stained, right? The red is the DNA and the protein and the RNA that make up the nuclear region, or the nucleoid. But you'll also sometimes hear it referred to as the nuclear body or the chromatin body. But I'll always stick to nucleoid as labeled in our diagram. What's the function? Well, DNA is the genetic information, right? Tells the cell how to do everything, when to do it. This is a bacterial cell that died, and actually this is its DNA rupturing out of the cell. Just poof, becoming part of the environment. Poor thing just exploded. So what structures have a similar function in eukaryotes? The nucleus. And I say structures because some of them will have more than one nucleus. Like paramecium have a macro and micronucleus. So the nucleus and a region within the nucleus known as the nucleolus are unique to eukaryotes, and as we know, that one of the defining characteristics. But similar functionality, right? We're still talking about storing genetic information. We're still talking about DNA and RNA and protein. The command center of the cell. And as I said, the nuclear envelope is actually 2 membranes. And within it, it has pores that allows information to leave, messenger RNA, right? And different messages to come in. And you see these circles here, right? These are the pores, the openings. This region in a cross section, and sometimes they'll have more than one region, so that would be nucleoli, right? If you just have one, it's the nucleolus. This is the production of ribosomal RNA that's happening in the nucleus. It's not membrane enclosed, so it's not an organelle. It's just a region within the nucleus. And as you can imagine, we make a lot of this, right? We make a lot of ribosomes. We have lots of ribosomes. So you're making it continually all the time, and so you tend to have this condensed region of ribosomal RNA within the nucleus. And so then that brings us to our beautiful little ribosomes, which aren't so little, right? They're really large molecular complexes. They're made out of protein and ribosomal RNA. So not just RNA, but protein as well. It helps provide the structural support. And their function is protein synthesis, to make proteins. Eukaryotes, all cells, whether they be eukaryote or prokaryote, have to have ribosomes, have to have the ability to make protein. The difference comes in the sizes of the ribosomes, the amino acid sequence within the protein that makes up the ribosome, and the RNA sequence. Because remember, how do we end up with the 2 domains, bacteria and archaea? Carl Woese studied what? Ribosomal RNA, right? He saw a distinctive difference among prokaryotes, so much so that they each got their own grouping. So ribosomes are made up of 2 subunits, a large and a small. And the system for measuring these is referred to as the Svinberg system. This takes into account the weight, but also you could say its aerodynamicness. So you can definitely tell that this one is small and this one's large because the number is small, 30, versus 50, right? But when you put the 2 of them together, the combined total isn't what we're used to seeing in traditional math, right? If you told your math teacher that 30 plus 50 is 70, she'd say wrong, right? Traditional adding, 30 plus 50 would be 80. But this system doesn't just take into account weight. So if we were just dealing with weights, right, say 30 grams and 50 grams, that should equal 80 grams, correct? This number together combined is larger, right, than the 2 separate. But it isn't what we expect, right? And that's because it's not just taking into account weight. It's also taking into account how this would move through a fluid matrix. Or you could say, almost think about, most of us understand resistance and aerodynamicness, right? Its structure affects how it moves through that medium. So it's going to move further because it's heavier. But again, this isn't just one thing you're taking into consideration, so you can't just add them together. The good news for you guys is only one number you need to memorize, and that 70S is the total size of the ribosomes found in prokaryotic cells. So this is both archaea and bacteria. And the reason for this is you will see this throughout your lifetime, especially if you continue on as a scientist. 70S is the total size in the Svinberg system of prokaryotic ribosomes, which remember prokaryotes are both bacteria and archaea. In contrast or comparison, do you think eukaryotes are bigger or smaller? They're bigger. Their subunits are 40 and 60, but their total that you need to know is 80. So if we were to look at the protein sequence, the amino acids that make up the proteins, they would be different for prokaryotes versus eukaryotes. And even when you get to the ribosomal RNA sequence, you're going to see differences between the two prokaryotes, archaea and bacteria. That's how we ended up with those two separate domains for those prokaryotes. And the really interesting thing here is, and one of the reasons why scientists believe that mitochondria and chloroplasts were once free-living prokaryotes, is that their ribosomes are prokaryotic. In our mitochondria, you have prokaryotic ribosomes and their own DNA. So now we're going to head on the outside. We're going to talk about external structures. So for prokaryotes, we're going to talk about capsules, Fimbriae, Flagella, and Pili. We're going to talk about how those bacterial flagella work, how they're arranged, because they'll sometimes have more than one, and talk about the rotation. In contrast, for eukaryotes, we'll look at their external structures of things like cilia and flagella. So both prokaryotes and eukaryotes can have, not all of them, that's why it says some, have flagella, and of course the function is the same. It's all about motility, locomotion, movement. Where we get differences is how they're structurally made and how they move. So for bacteria or prokaryotes, their flagella is made up of a single protein known as flagellin. And these proteins stack up on themselves. The movement of the flagella is due to proton motive force. Eukaryotes, on the other hand, their flagella are made of microtubules, typically in a particular pattern. So you'll notice that they tend to have 2 central microtubules and then 9 pairs around the outside. So this is sometimes referred to as the 9-to-2 pattern. So they're more complex, and they don't move, they don't rotate. They wave back and forth. So what structures similar to flagella do eukaryotes have that prokaryotes don't have? Cilia, right? And cilia are just shortened flagella. So here's a cute little paramecium, right? Just covered in cilia. This is a flagella right here, and this one is a little bit different. It's got additional proteins sticking out from it. Why would that be beneficial? Why would you want flagella with like all these little hairs on it? You'd move faster because, again, you've got a greater surface area, right? So this is equated to someone sticks you in a canoe with a little paddle or a big paddle. Which one you picking? I'm doing the big paddle, right? You'll get there faster. Right? You can push more water and you can get there faster. So for prokaryotes, they will have sometimes more than one flagella. Like this guy, he's completely covered in flagella. So you see all these little hair-like structures? That's all flagella. He's covered the entire surface. So it's like scary cousin It from the Addams Family. The terminology that we use for this flagellar arrangement, having completely covered in flagella, is referred to as peritrichous. Trichous means hair-like, peri means many. This guy, on the other hand, has one single flagella. So that one is referred to as monotrichous. Mono meaning one. You'll also sometimes see it referred to as being polar, because notice it's at one end, right? The poles are at the different ends of the Earth. What's the shape of these two bacteria? Rod-shaped or bacillus. What's the shape of the two down here? Spirillum. They're not spirochetes. Spirochetes would be a tighter coil. They almost look like zigzag lines under a light microscope. These are probably electron micrographs. These are pretty detailed. These are probably light microscope pictures, and these have probably been stained with the special stain that enhances the flagella, right? So it makes it appear thicker than it is. The next term, Lophotrichous, refers to a clustering of flagella at one end, or in the case of this picture, both ends. Do you see how there's multiple flagella at both ends? But this term just means a clustering at an end. So you could have an organism that has a cluster just at one end and not at both, like you see here. Now, where do you see flagella for this next one? You see one at each end, right? So this is amphi, meaning opposite, right? So one at each end, at each opposite end. We didn't see any cocci here, did we? I don't know, do you see any round boats out there either? Not usually. They don't move really well. There are a few cocci out there that have flagella. They don't move so well, though, right? So it makes sense, like a rod or something that's just a longer rod would be modal as opposed to something that's round. Most of the cocci we deal with are non-modal. Round boats just don't cut it. Papillae, there can be two types, and these are protein appendages outside the cell, and these are for prokaryotes. If that appendage hooks up with another cell to be able to exchange genetic information, one of the most common things they'll exchange are those plasmids that I talked about, right? Where they'll give that DNA to another cell. They join up via this protein hollow tube known as a sex papillus, or sometimes the fertility papillus. This is sometimes referred to as bacterial sex. The proper term is conjugation. Because in truth, all sex is an exchange of genetic information. Bacteria are not required to do this, though, unlike eukaryotes like ourselves, right, where we actually have to combine our genetic information to produce another organism. Bacteria can reproduce asexually, but they can occasionally share genetic information. They can conjugate with other cells, and they do that via the sex papillus. Right? They literally have a hollow tube that joins the two cells. Fimbriae, on the other hand, is when those protein projections tend to be smaller, and they tend to have a lot more of them. And this can aid in movement for some organisms. They can almost like twitch. But for the most part, it's used for attachment, right? To actually be able to attach to surfaces. So, for example, gonorrhea, Neisseria gonorrhea that causes the infection known as gonorrhea, its papillae are what the specialized papillae we call fimbriae is what allows it to attach to our epithelial cells, right? So those proteins are grabbing onto proteins on our cells and are why we can get that infection, because they can stick to us. So we're like, oh, wow, we should just make a vaccine against the papillae, right? Against those fimbriae. Then we'd be all set. We'd never get gonorrhea. Wouldn't that be awesome? Why haven't we done it yet? Because those papillae, the fimbriae, they're changing them all the time. It doesn't stay exactly the same. This is why you don't get immunity to gonorrhea either, and why you can get it multiple times. Because you can't develop antibodies against it, because the gonorrhea you get the first time is probably different from the gonorrhea you got the second time. What's the really good news about this? Everybody in this room should know how you get that disease, right? It's sexually transmitted. It can be avoided. The other good news is it's a bacterial infection, so what can you take to cure it? Antibiotics. What's the bad news as it relates to antibiotics in this world we live in? There is resistance out there. There are some strains of the bacteria that are resistant to the commonly used antibiotics against the infection. Don't look at the CDC's website. It will terrify you. Although gonorrhea isn't the leading sexually transmitted disease. Anyone know who's number one? Nah, well, probably, but that one kind of doesn't count. Everybody pretty much has herpes. Chlamydia. Because herpes you can get other than sexually transmitted, right? Because you can get cold sore herpes, right? And, you don't have to have sex to do that. You just have to kiss somebody. Chlamydia. 1 million cases each year in the United States. And Louisiana is one of the leading states in the country when it comes to sexually transmitted diseases. Yeah. Be very choosy. Very choosy. Okay. Make sense? All right. So capsules and slime layers. So this is outside the cell wall or the cell membranes. And this is an additional layers that can add, of course, additional protection. In some cases, it can add and enable attachment. Again, give that bacteria the ability to stick to us. And only with slime can it also be used as something that's motile, right? That slime can help it be motile. So what's the difference structurally between capsules and slime layers? So capsules, as you can see in this picture, so here's a bacteria, and you see this clear area around the bacteria? That's its capsule. They've done a special stain where they've stained the background and the cell, but capsules don't have a charge. They don't stain. So they appear as these clear halos around the outside of the cell, because it's its outermost covering if it has it. So this is outside the cell wall, outside the cell membrane. What can you say about that layer? It's pretty uniform, huh? The thickness is pretty much the same all the way around. Same thing here. This is actually a yeast, right? So if this is a yeast, do eukaryotes have capsules? What's a yeast? It's a fungus, right? What are fungi? Eukaryotes. So the answer is yes. Although very common in, oops, I'm losing it. Though very common in bacteria, because it aids in attachment and helps with protection, you have some eukaryotes, especially yeast, that produce capsules. This is actually 2 cells. You see the 1, 2, they just divided. But this clear region is the capsule. This is a little bit different staining procedure. This is called India ink. I don't like this one because it's little droplets of black dye, and in a liquid, they move, right? They vibrate because of Brownian motion, the normal heat that exists and causes water molecules to collide with one another. So like if you look at this one for a long period of time and it's vibrating, it gives me a headache. I don't like that stain. I don't do that stain anymore. We do dye stains, different dyes like this for the bacteria and for the yeast that we work with in lab. So this is a summary of that chart that I started out with right at the beginning and the end of the other lecture. So the prokaryotic cytoskeleton are long, thin filaments. As you saw in that one picture, the green lines, they're made out of protein. And they do similar things that eukaryotes do. They're going to help with cell division and localize proteins and help with cell shape. Inclusion bodies, remember, are only found for prokaryotes. These are areas within the cytoplasm. They can be enclosed, right, and contain a large amount of the same material, right? So they'll tend to pack all the same things in the same spot. Their shape can be circular or even oblong, and that really depends on how it's enclosed. Is it enclosed in protein or phospholipid proteins, and what's in it? Is it organic or is it inorganic? And the function, of course, is storage and reduce osmotic pressure. Plasmids, not found in all prokaryotes, right, but it's very common. And some eukaryotes, we've seen it. It's commonly circular, but it can be linear, and it's extra chromosome, right, so extra DNA. And so therefore it's an extra advantage if they have it, right? More information. You can do more things with more information. The nucleoid is only in prokaryotes, and it's irregularly shaped, as you saw in that picture, that red zone. It's the DNA, the RNA, and the protein that make up basically the chromosome. And it stores, of course, genetic information. Ribosomes, all prokaryotes and eukaryotes have to have ribosomes. They have to be able to make proteins. All cells have to have ribosomes. But remember, there are differences. What's the differences between prokaryotes and eukaryotes? Size, yep. What are prokaryotes? 70. Eukaryotes? ADS. It's Fimbriae system of measuring. Is their ribosomal RNA the same? No, even within prokaryotes, you've got two differences. What two domains of prokaryotes? Bacteria and archaea. And then eukaryotes are distinctly different in their ribosomal RNA. Even their protein is different in these ribosomes. Because remember, ribosomes are made of ribosomal RNA and protein. So, endospores. These can only be formed by some bacteria. And we learned about a disease on our selected disease list that can form endospores. And that's why it's kind of scary. Which one? It was sent as a powder, millions upon millions, yeah, anthrax, of spores. So unfortunately, the definitions sound like reproduction, right? So the process of forming the spore is sporulization, right? When it comes back out of the spore, we call it germination. The reasoning for this is probably at one time they thought it was a form of reproduction, but in fact it is not, right? It's not like what we think of spores like from molds, right? This is just a dormancy period for these cells. And it forms within the cell. That's why it's called an endospore. Although some scientists like myself are bad sometimes and we just say spore. We shorten it. But when we're talking about bacteria, we're talking about endospores. So there are two main genera, or genuses, right, of organisms that you'll run into that produce endospores. So one of them is what anthrax is. Do you guys remember what the genus name for anthrax? Bacillus. 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 So they're what shape? Rod-shaped. Just as you see here. So again, not a form of reproduction. So when are they going to do this? Why would you go dormant? Why would you stop metabolizing, stop reproducing? There's no food, right? Like bears hibernate in the winter because there's no food, right? So if you run out of food or oxygen or carbon dioxide or water or anything necessary for life, then instead of dying, these cells go dormant. And what are you going to protect? What's the most precious thing in your cell that you must protect to survive? The DNA. So what they do is they encase and stabilize the DNA. And of course this is within the cell. So they form layers around the DNA, a copy of the DNA, and they sequester it and stabilize it so that when conditions become harsh, they can just wait it out. Then when conditions become good again, it turns back into the original cell it once was. And that process is called germination. So since it forms within the cell, that's why it's called an endospore. Eventually the cell itself will usually deteriorate, and therefore you have the spore all by itself, what we call a free spore. So we already named one of the genuses. What was that? Bacillus. The other is probably one you've heard of, and that's Clostridium. And one that's not so well heard of is Sporosarcinia, which actually happens to be a cocci. Clostridium, just like Bacillus, is rod-shaped. Anybody know any Clostridium that are spore formers that can cause disease? We vaccinate ourselves every 10 years against this one, against the toxin it produces, because it can cause things like your jaw to lock up. Tetanus. Clostridium tetani, right? It's a spore former. It's found throughout the environment in the soil. You really can't avoid this one. And the bad news, again, is the toxin it produces. So we actually vaccinate ourselves against the toxin and not the bacteria itself. So the vaccine won't stop you from getting the infection, but will stop the debilitating effects of the toxin itself. You'd have to take antibiotics to get rid of the infection. So since most of the bacteria that form endospores are rod-shaped, their location that they always form their spore in is always the same. So this helps in identification too. So if that spore always forms right smack in the middle, we say that that spore's location is central. If it always forms way at the end of the cell, it's referred to as terminal. If it's somewhere between the central and terminal end, it's referred to as subterminal, right? It's not at the end, it's not in the middle, it's close to the end. And sometimes the spore is much larger than the cell itself, will even cause the cell to look swollen where the spore has formed. Sporangium is the name sometimes given to the cell when it has a spore inside of it, or sometimes the spore mother cell. And again, these sounds like reproduction, right? But they're not. It's not reproduction. So we have an animation for this. Let me plug it in. And for these animations, sometimes they're going to go in a little bit more detail than I'm going to hold you guys to, right? So just the basics that we just went over as it relates to endospores. Let's see if I can get it to play. So back to Canvas. And we'll do spore formation. And we will skip that.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing spore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Picolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
So it's all about protecting that DNA. And notice in the process, it's actually drying it out, right? Water leaves. So a spore is already dry to begin with, so you can't dry it out to destroy it, right? Most chemicals can't penetrate that spore coat. UV light can't penetrate it. Boiling won't destroy it. The only way you can usually get rid of it is high pressure steam in an autoclave, basically a pressure cooker. And that's one of the main reasons why we use autoclaves for sterilization, is it will actually destroy endospores. Oh, let me play the motility one. So locomotion, for the most part, right, is by flagella. So we're going to watch. Wait a minute.
Hi, I'm John, and I can save 1 million lives with just refrigerant.
Most motile bacteria move by means of flagella. A flagellum is a slender structure about 15 to 20 microns long and about 20 nanometers wide. A typical rod-shaped bacterial cell is 2 to 3 microns in length. The bacterial flagellum is composed of 3 parts: the basal body, which is embedded in the cell membrane and wall, a short curved segment called the hook, and the portion that extends out from the cell called the filament. The basal body consists of rings that correspond to the layers of the cell envelope, and therefore differ in gram-negative and gram-positive bacteria. In most gram-negative bacteria, there are 4 rings connected to a central rod. Gram-positive bacteria have only 2 basal body rings, one connected to the cell membrane and the other to the cell wall. Motility is mediated by proteins associated with the plasma membrane and basal body, and is powered by the proton motive force associated with the plasma membrane. The filament is a rigid structure which rotates to move the bacterial cell. Spirochetes are motile by means of bundles of flagella, called axial fibrils, that run along the cell within the periplasmic space. The number of fibrils varies. These axial fibrils extend from both ends of the cell within a flexible outer sheath. Spirochetes have a screw-like motion which allows them to bore into mucoid layers and soft agar media. Some bacteria lack flagellum or axial fibrils, but are motile if they are in contact with a solid surface. This type of movement is referred to as gliding motility. For some gliding bacteria, secretion of a slimy polysaccharide appears to be involved. Other bacteria appear to have special proteins on their surfaces that mediate gliding motility.
So this next one, you'll notice the note that I say just watch
When Escherichia coli moves in a medium...
Just watch the introduction, because then it starts getting really detailed, literally down to the molecular level of how this does. So if you're having trouble sleep, maybe watch the second half.
That lacks a concentration gradient. The cell travels, stops, or tumbles, and then continues moving in a new random direction. When the flagella rotate counterclockwise, the flagella form a tight bundle and propel the cell forward in a run. After a brief period, the direction of rotation is reversed, causing a tumble. As cells move up a chemical gradient, the runs are longer than when they travel down the gradient. The overall result is random movement in the absence of a chemical gradient, and movement toward a chemical when a gradient exists. Attractant chemicals bind to chemoreceptors called methyl-accepting chemotaxis proteins, located in the cell membrane. The cytoplasmic side of the MCP interacts with 2 proteins, KW and Ka. When the MCP is not bound to an attractant, it stimulates Ka to phosphorylate itself using ATP. Ka autophosphorylation is inhibited when the attractant is bound to its MCP.
Who's asleep?
Phosphorylated Ka can donate.
Yeah, it just keeps going. The important part is right here in the beginning. How does that bacteria know there's something it likes or doesn't like in the environment? Chemoreceptors, right? Proteins in its membrane. They don't have eyes and ears like we do, right? Can't smell that something is, or noses. Can't smell something's good, right? Can't smell bacon cooking in the kitchen, right? Instead, they have proteins that are their receptors in their cell walls, in membranes. That's going to help them sense, okay, is this good? Do I want to go that way? Or is that bad and I'm going to run away? And then you notice that like E. coli is completely covered. What's the term for completely covered in flagella? Peritrichous, right? Yeah, peritrichous. And you notice too, like he orients all of the flagella in one direction when he's running, right? All his little hairs. It's like, imagine like we're running and phew, our hair's flying back, right? But when he tumbles, it's like all over the place, right? And his flagella, remember, rotate, but it's that hook, right, that creates that propulsion. So just like a propeller on a boat, right? Think about how the propeller rotates and propels the boat down the bayou, right? So the same thing for these guys. That's how their flagella work. They rotate. So all we have is, so here, that animation, you know, they tried, right? But the spirochetes didn't look so cool, did they? So I found a YouTube video. You can actually see actual spirochetes moving. So check them out. Isn't that cool? Like, you're going through. Cooler than a snake, I think. Alright. So that's a little short fun. Let me minimize that. Okay. Almost done, y'all. So when we talk about bacteria and motility, we definitely, number one, we always think of flagella. But what else did you see in that other video? Or the one we just watched. What are they doing? They're corkscrewing, right? So only spirochetes do that, though. Some have flagella, and they can move by flagella, by it rotating. What else did we see? The slime one, right? The gliding motility, and usually due to slime. And then one other one, they kind of twitched because of the proteins, but we actually have a name for those proteins now. Starts with an F. Fimbriae, right? So we've got flagella moves like a propeller, right? They rotate. Spirochetes, that corkscrew movement due to their axial filaments. Gliding, a lot of times due to slime production. And that twitching via Fimbriae. So how do the bacteria use their locomotive ability to swim towards attractants and away from repellents? What do we say? How do they know? The chemoreceptors, those proteins embedded in their membranes. So we already watched the animation, but again, this is just showing you that when he's running, right, especially if they're covered in flagella, they kind of all orient in one direction. They can actually turn those discs in the membrane and cell wall and orient so that they're all going in one direction. But when they tumble, this is why they don't turn like a car does. Right, they just tumble around. They're not very good at turning. Right, they're not good at changing direction. They just tumble around. So the last is somewhat of a review, right, from and what we talked about in the beginning, all the different organelles and how they function. Should be a review from biology. And then endosymbiotic theory, can't remember if I wrote it out, is that belief that the mitochondria and the chloroplast were once free-living prokaryotes engulfed by an early eukaryote, and they began that symbiotic relationship, so much so that they can't exist without each other anymore. And a lot of the evidence to that is that both mitochondria, which are our powerhouses, right, for our cells, that's what creates the energy, the ATP, or photosynthesis and photosynthetic organisms, is that both of these have their own ribosomes and they are prokaryotic. And they have DNA that's very reminiscent of prokaryotes as opposed to eukaryotes. So what are the two main organelles for energy? Mitochondria and chloroplast. Mitochondria is chemical energy, right? Where chloroplasts allow them to do photosynthesis, light energy, to create molecules. Plants actually have both mitochondria and chloroplasts. We, on the other hand, only have mitochondria. If we had chloroplasts, we could tell our moms that we don't have to eat our vegetables, and we'd probably look like green Martians. Yeah, the mitochondria and the chloroplasts. So this is unique to eukaryotes, this ability to engulf stuff inside the cell. If the cell surrounds its membrane around something and engulfs it in such a way to, say, digest it, say if it's a white blood cell in our bodies and we engulf bacteria or viruses to destroy them, that process is referred to as phagocytosis, basically to eat. Endocytosis is actually a general term to engulfing stuff and bringing it within. Autophagy, auto means what? Self. And so this is where we're engulfing and digesting our own cellular components. And this is one of the ways that we'll recycle the components of worn out mitochondria. So lysosomes are very key to these processes because they contain digestive enzymes. So the process in which lysosomes are made are very important to the biosynthetic secretory as well as endocytotic pathways. So we'll watch one last animation. I wonder if this is even a link. No, I wasn't that fancy. Okay. So the last one, lysosome formation. Let's see if I can get a play in here.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
And that's referred to as autophagy, like when we basically eat ourselves. So what was involved? Where did it all start? Where were those enzymes made? Yep, the endoplasmic reticulum, but which one? The rough endoplasmic reticulum, the one coated in ribosomes. And then that vesicle traveled to what other organelle? The Golgi apparatus. And then we actually had the lysosome. I always think that my nickname that I give to the Golgi apparatus is the packing plant. You know, he kind of puts everything together, puts the final touches to be able to send it on its way. And so stuff that actually leaves your cell actually has to be processed through basically the packing plant, the Golgi apparatus, before that membrane would fuse with our membrane and those substances would leave your cell. In the case of lysosomes, we pretty much keep them within the cell. We can have them fuse with, do I have a bigger picture? Yeah. We can have them fuse with the phagosome, right, when phagocytic cells engulf things like bacteria, and then the enzymes in the lysosome will fuse with it and digest it. Or we can fuse it with organelles and digest them like mitochondria. That's autophagy. And even large substances we can take in vesicles via just the general process of endocytosis. And if it needs to be digested, we're going to fuse it with a lysosome to do that. And again, these are destructive enzymes. We don't want them running free willy-nilly throughout the cell and digesting anything they want. One of the ways we control them is keeping them in this vesicle, in that membrane, what we call lysosomes, right? So this is a pretty good summary table, except I've lined out. They go into a little bit more detail in this than we did in the course, so obviously I don't expect you guys to know that. But remember, again, as I show here, when we talk about prokaryotes, we're talking about bacteria and archaea. And so what you really want to focus in on, of course, is the differences. So remember, those membranes are distinctly different in their bonding and what they're made out of. Flagella are teeny tiny, submicroscopic, and made of just one protein fiber for all prokaryotes. Where eukaryotes, as I said, you have those microtubules in that 9:2 pattern. Endoplasmic reticulum, Golgi apparatus, these are organelles, right? So we expect to only find these in eukaryotes, in the domain eukarya. Again, notice the size of the ribosomes and the fact that only peptidoglycan is found in bacteria, right? That's a defining characteristic of bacteria. So they have gas vesicles written here, but that's an example of an inclusion body. So again, that would be prokaryotic, not eukaryote. In eukaryotes, you would have lysosomes and different types of vacuoles that are actually membrane encased. And again, mitochondria, chloroplasts, those are organelles, so only eukaryotes. And nucleolus, what's in the nucleolus? What's happening in that region? Ribosomal RNA synthesis. And again, that's only a region found in eukaryotes within their nucleus.